Hello, I'm Vladimir Sedivy. I'm a consultant for Solar Saltworks, and we are here at one of the largest solar saltworks in Europe. I'm going to talk to you about the biology of solar saltworks, how important it is for the environment and how important it is also for the solar salt production. We are here at the intake of the seawater. There is a pumping station for seawater that brings the fresh, clean seawater from the Mediterranean Sea to these channels here. Through this channel here, the seawater is pumped to a series of ponds, where from one pond to the other the concentration of the seawater is rising, until it reaches the concentration when the sea salt begins to crystallize. Crystallization of salt is a physical process, but to understand why well-managed biological system in solar saltworks is favorizing this physical crystallization, let me first take you to the sea. 70% of our Earth is covered by the sea. On average, seawater has a salinity of about 3.5%. Apart from salt, seawater also contains nutrients such as nitrogen and phosphorus. Plants that live in water need these nutrients to grow. With the power of photosynthesis, sunlight transforms water and carbon dioxide into sugar and oxygen. Sea creatures take in oxygen and feed on plants. Oxygen and sugars are converted into movement and carbon dioxide, and nutrients in turn are released. Small animals get eaten by larger ones. They live, thrive, and eventually die. Due to gravity, this leftover organic material sinks to the bottom of the ocean, where microbes feed on the tissues. The nutrients that were fixed by the plants at the water surface are released again at depth by the activity of microbes. Some of the organic matter is transformed to carbon dioxide and other gases. Thanks to physical processes such as storms, wind, or the great ocean current, the nutrients are brought back to the water surface, where they are consumed by the plants again, completing the nutrient cycle. In solar saltworks, we have a different situation. Here the brine flows from one pond to the other and evaporates due to solar radiation and wind. This results in rising concentrations of salinity. In the first concentration pond, salinity is close to seawater, and in the last concentration pond it becomes saturated at 26 degrees Beaumont. As the salinity increases, so does the concentration of nutrients in the water, if they are not consumed by plants. Some plants and animals have adapted to live in bodies of water with higher salinities, such as in hypersaline wetlands or salt fields. In the high salinity water we find the halophil microalga Dunaliella salina. The higher the concentration of nutrients, the faster the alga grows. Dunaliella salina is a plant. It absorbs light, carbon dioxide and nutrients and produces sugar and oxygen.
Equally well adapted to high salinities is the brine shrimp Artemia salina. Dunaliella salina is their favorite food. Artemia releases the non-digested food residuum in form of fecal pellets to the pond bottom. The fecal pellets contain the non-digested nitrogen and phosphorus. Now this is where it gets especially interesting for people looking to produce high-quality salt. Benthic mats at the bottom of the pond overgrow the fecal pellets, sequestering the nutrients. Additionally, the mats create a physical barrier, reducing brine losses through seepage. The dark mats also increase the absorption of solar radiation and thus increase evaporation. This process takes place in every pond until the biologically purified, nutrient-free and saturated seawater flows to the salt crystallizers. As a result of these biological processes, this brine contains no organic material that could interfere with sodium chloride crystallization. In the crystallizers, the brine further evaporates and the salt grows. The crystals are large, hard, transparent, and very pure. So far, so good. But this works only if the salinity in any given pond is constant and if the change of salinity between the subsequent ponds is small. Dunaliella salina, Artemia salina, and the benthic mats can only strive in constant salinities. If the concentration change between the ponds is large, the planktonic biota flowing from one pond to the next perishes and decomposes. The nutrients proceed to the crystallizers where they support algal bloom. As a result, mucilage is formed, which interferes with the sodium chloride crystallization. Instead of beautiful large crystals, you get small agglomerates which are non-upgradable because they are soaked with bitterness. As a general rule of thumb, the salinity difference between two subsequent ponds should be about 1 degree Baume, and the optimal number of ponds is 24.
Unfortunately, many salt works, they don't consider biology to be important for making salt. It's a pity, because with modern technologies and with good understanding of the biological processes in solar salt works, it is possible to make a very good quality salt suitable for human consumption, but also for the much more demanding chemical industry using membrane cells at significantly lower cost and higher quality. Thank you very much.